The overall goal of the following experiment is to test whether a particular DNA variant, such as one associated with a disease, has an effect on the expression of a particular gene. This is achieved by selecting cell lines that are heterozygous for both this variant and a coding SNP in the gene of interest. As a second step, DNA and RNA are extracted from the cells and then used as template for an allele-specific primary extension reaction. Next, primary extension products are analyzed by mass spectrometry in order to quantify the relative abundance of the two alleles. Results are obtained that show whether the original DNA variant associated with a disease is also associated with a difference in gene expression based on quantitative allele-specific measurements by mass spectrometry.
The main advantages of this technique are that, unlike with a luciferase reporter gene assay, it doesn't require molecular manipulation and it measures endogenous levels of gene expression. Moreover, we're able to compare levels of expression between the two alleles in an internally controlled way, which removes some of the experimental variability.
This method helps us answer key questions in the field of human genetics, such as the molecular mechanisms underlying genetic associations.
We first had the idea of this method when we identified a genetic association spanning 3 genes and could not refine the signal to a single one by further genetic mapping.
The cells used for this assay should express the gene of interest, as well as meet 2 fundamental genotypic requirements. They have to be heterozygous both for the DNA sequences associated with disease and for DNA markers, which can distinguish transcripts based on their chromosomal origin. In the cell line selection strategy shown here, cell lines A and B are heterozygous for a transcribed coding marker, represented by triangles. However, only cell line A is heterozygous for the DNA sequence associated with disease, called the risk DNA variant, represented by circles. The blue allele of the risk variant is associated with a lower level of transcription. After selecting the cell line, culture the cells in a flask according to the supplier's specifications. We will use fibroblasts in this demonstration, although this assay is equally applicable to other cell types, including primary cells. Prepare 2 10 cm Petri dishes for DNA and RNA extractions respectively. When cells reach 80% confluence, they can be harvested for DNA and RNA extractions using standard methods, including commercially available kits that are appropriate to the cell type selected. Prior to performing this assay, design 2 pairs of PCR primers for each DNA marker, 1 pair for genomic DNA amplification and the other for cDNA amplification. Place forward and reverse primers for cDNA amplification in different exons to avoid genomic contamination. Design primers to obtain a PCR product in the range of 100 to 500 base pairs in length. For each cell line, prepare 8 10 microliter PCR reactions, 4 independent reactions for genomic DNA amplification and 4 independent reactions for cDNA amplification. Each 10 microliter PCR reaction contains allele TAC, dNTPs, primers, magnesium chloride, and either genomic DNA or cDNA. Run the PCR reactions under the conditions optimized for each primer pair, keeping the cycle number in the linear phase of amplification. Once the PCR reactions are complete, remove PCR primers and excess dNTPs by adding exonuclease 1 and shrimp alkaline phosphatase, SAP, to each PCR product and incubating at 37 degrees Celsius for 20 minutes, followed by 15 minutes at 85 degrees Celsius. Spot each PCR product twice onto a 384 well plate, so that 8 measurements will be available for each sample, 16 spots in total. For the primer extension assay, design the extension primer with the assay design software so that the same primer can be used for both genomic and cDNA PCR products. Specify primer length in the range of 14 to 18 base pairs. Carry out the primer extension reaction with the appropriate mix of 3 dideoxynucleotide terminators and 1 deoxynucleotide. A typical primer extension reaction includes a starting step at 94 degrees Celsius for 2 minutes, followed by 40 cycles of 94 degrees Celsius for 5 seconds, 52 degrees Celsius for 5 seconds, and 72 degrees Celsius for 5 seconds. Finally, the extended oligonucleotides are quantified by matrix-assisted laser desorption ionization time-of-flight mass spectrometry using a spectroreader mass spectrometer.
A key point of this protocol is to use several strategies to control for experimental noise. To rule out artifacts, it's really important to repeat the experiment at least 3 times.
The best control for the experiment is using a cell line heterozygous for the coding SNP, assayed for primer extension, but that does not carry the disease-associated allele. Additionally, design assays for alternative DNA transcribed coding markers. If these are not available, an additional control can be provided by alternative PCR primer pairs targeting different exons. For the primer extension assay, an internal control can be provided by the design of extension primers annealing to both forward and reverse strands. An example of allele-specific expression analysis is shown here with examples of mass spectrometry traces. This figure shows the results from the analysis of 4 allele-specific primer extension products carried out on 4 different templates. The top 2 graphs represent the results obtained from the analysis of genomic DNA of 2 different cell lines, both of which are heterozygous for a transcribed coding polymorphism. However, only cell line A is heterozygous for the risk DNA variant. These lower 2 graphs represent the results obtained from the analysis of the cDNA generated from the same cell lines. As a result of experimental artifact, the first peak is always higher than the second peak, even in the genomic analysis. Therefore, the results from the genomic analysis are used to normalize the cDNA data. Statistical analysis is conducted by extracting the allelotyping report within the mass array typer software. This report provides data about the peak area, and we show here an example relative to 2 cell lines, cell A and cell B, heterozygous for a coding SNP, but where only cell A is heterozygous for a disease-associated marker. The columns labelled in grey are directly generated by the software, while columns labelled in green are derived manually. For each cell, there are 8 measurements for both the cDNA and the genomic DNA. The software will calculate the area underneath each spectrum peak, which represents the abundance of each of the 2 alleles. The allele ratio is calculated by dividing the area of allele 1 by the area of the allele 2. For each cDNA and genomic sample, a mean and standard error are calculated across the 8 measurements. A high standard error, greater than 0.1, indicates that measurements are not consistent and should be disregarded. Lastly, the cDNA mean ratio is normalized using the genomic DNA mean ratio to estimate the deviation from a ratio of 1, which is expected when there is no difference in allele-specific expression. The normalized ratio is different from 1 in cell A, but close to 1 in cell B. The data suggests that cell line A carries a DNA variant in phase with the allele measured by the second peak, which reduces the expression of the gene under analysis. Cell line B provides a very convenient negative control.
While attempting this procedure, it's really important to define informative genetic variants and to define appropriate experimental controls.
Following this procedure, other methods can be used, such as haplotype-specific chromatin immunoprecipitation, which help you answer questions such as where there are genetic variants for which there are no transcribed markers to use to analyse allele-specific gene expression.
After watching this video, you should have a good understanding on how to analyze RNA in an allele-specific manner and how to use this to understand genetically important functional variants.